So far, I've been talking almost exclusively about either the communication within a single neuron, the way an action potential is going to go down a neuron, or the communication between two neurons, from the presynaptic neuron through the synaptic cleft to the postsynaptic neuron in terms of neurotransmitters. But I want to stop and emphasize that single neurons or even single pathways between a line of neurons don't really tell us that much about what the stimulation in our brains is actually doing, because the communication networks in your brain are so much more complex than this. Think of one, again, remember the 10,000 connections that neurons might have and the combinations of cascading information that might go through those really complex combinations and pathways. So behaviors, feelings, thoughts, the way you move, what you're saying, none of those are happening from a single neuron being activated or a single message even. They are through very complex patterns of activation within your brain and through your body. These are groups of neurons firing in very specific combinations that, when repeated, they might lead us to be able to have repeated memories, experiences, thoughts, behaviors, but there are these very complex patterns through the brain, not just like one brain cell firing at a time. So obviously, we can't really study this very completely or directly because it is so complicated. So we can study this a little bit better in very simple organisms, like brains that, or organisms that don't even have brains, for example, to see how this works. And we're starting to be able to use computer simulations to study this. But it's not like we can point to a brain or point to a neuron and say, hey, look, there's where this thought is happening, because it's happening throughout a vast network of signals and communication. Another system that I've already mentioned that I'll bring it back to is the nervous system. Remember, when I'm talking about neurons, they make up this nervous system that we have that communicates throughout our body. Remember, I said the central nervous system is the brain and the spinal cord. And the peripheral nervous system is all the other nerves and neurons in your body. Those different sensory neurons at the periphery of your body, the glial cells holding everything up, all that other communication is that peripheral nervous system. So you might think about this broken down in this way: the central nervous system on one side, peripheral on the other. Um, Remember, the brain and spinal cord are part of the CNS. The peripheral nervous system has its own parts too. The somatic nervous system, the things that are voluntary, like when I move parts of my body, that's the somatic nervous system working. But we have tons and tons of automatic functions too that are part of the autonomic nervous system that's involuntary. And the autonomic nervous system involves two different processes as well, or two different systems that excite us or arouse us and get our bodies moving, or the parasympathetic nervous system, things that calm us down. So, like your fight or flight response, we'll talk about in a bit, is your sympathetic nervous system getting activated so you can escape danger or fight back against something. And the sympathetic nervous system would be the opposite. It's calming down your body, diverting energy to things like digestion or reproduction. So all these things kind of work in concert to keep your body functioning properly. So to talk about these things, some of them more specifically, I want to spend some time on the brain, and I want you to think about this part of the lesson as just familiarizing yourself with different parts of the brain. I'm going to assume that there are people that have never thought about or heard about the different parts of the brain. There are other classes at Hamlin that you would be able to take that will tell you much more about these specific pieces. But we just have, this week and this lecture really to talk about this stuff. So you might think about the different parts of the brain in several ways. Maybe the broadest way to separate out the different parts would be the forebrain, the midbrain, and the hindbrain. Now, the hindbrain is the most primitive or simple part of the brain. That's where things like your involuntary movements and stasis functions like breathing and keeping your heart beating and stuff are organized. So that's going on in this part. And we have, relative to the whole size of our brain, a relatively small hindbrain. Lower animals, mice, fish, stuff like that, they have much bigger hindbrains relative to their forebrain. The forebrain is what makes us complex and capable of, complex thoughts and executive function and all these things that make us human and nuanced and things like that. That's why our forebrain is so big. You'll notice in lower animals, the forebrain is much, much smaller. And the midbrain basically takes the role of, well, one of its many roles is communicating between these more modern functions and these more primitive functions and making sure they kind of work in concert. If we break this up in a different way, you can imagine this brain kind of sliced down the middle, and you're looking in at one side of it. We can see these inner parts of the midbrain and these two different parts of the hindbrain. The brain stem controlling a lot of involuntary functions, and the cerebellum here controlling things like motor function. And you have all these different parts inside here. The hypothalamus controlling a lot of hormones and signals, along with the pituitary gland. The amygdala has to do with emotion. The hippocampus has a lot to do with memory. We'll circle back to talk about the hippocampus quite a bit when we get to memory. And other parts of the brain, like the cerebral cortex, again, that part with all the folds and all of our spreading activation through neurons, where we can do complex thoughts and coordinate complex actions and things like that. This other part here, this bundle of fibers here, the corpus callosum, this is where the two hemispheres of your brain are connected, basically. And we'll talk about in sensation and perception what that has to do with how we know about the different functions that are distributed across the different hemispheres. There are some things like language, for example, that are more associated with your left hemisphere than your right hemisphere, and the ways that they communicate across the brain can be really interesting. And if you have patients that have a severed corpus callosum, for example, there might be ways that we can use that unfortunate sort of injury or surgery to tell us something about that communication between the hemispheres. So if we then look at the outside of the brain again, I have this kind of false color image showing specifically the cerebral cortex and some of the different parts of it. I've talked a little bit about these parts of the hindbrain here, the brainstem and cerebellum, but these are the more sort of complex things that make us smart, I guess. The frontal lobe is where a lot of our problem solving, executive functioning, understanding what's going on, producing speech, all that's going on here in our frontal lobe. Our temporal lobe has to do with understanding language, has a lot to do with memory, and it's kind of next to your ears. So that's one way to remember that it has to do with your auditory processing as well. In the back of your brain is the occipital lobe, which kind of funny that it's the furthest away from your eyes, but this is where your vision is processed and controlled. We'll talk in sensation and perception, how your nerve from your eyes goes straight back through your brain into the occipital lobe. This is where you're processing all the things that you're seeing and understanding how that works, and we'll touch on that quite a bit. Finally, the parietal lobe has a lot to do with your body orientation. Generally, we might sometimes call this your somatosensory cortex. It's how much you're feeling from those sensory neurons, and things are represented in your parietal lobe kind of proportional to how much feeling you get from them. So your fingertips, for example, take up an inordinate amount of space here compared to the rest of your body. Where your like right hip, for example, you probably don't have a lot of space here devoted to that, but you have a lot of space devoted to your lips, your fingertips, things like that have more sensory input to them. So we know these things with some confidence, but how do we actually know the different parts of the brain and how they function? If we actually just to go back to the metaphor I used in the first lectures of the black box, if we look inside the black box, there's only so much we can do by like popping open someone's head and looking at the pieces of their brain and looking at the different neurons there. That doesn't really tell us how the machine works. So dissecting a person after they're dead doesn't really do that much for us. Another thing we can do is we can sort of use unfortunate accidents really to tell what damage to different parts of the brain might do. So if someone damages a particular part of their brain through an injury or an aneurysm or something, and they lose a particular ability, then that might tell us, well, that must be a part of the brain that does X. So if you lose your problem solving ability, because you have an injury to your frontal cortex, then we might think, well, that must be where we're doing a lot of our information processing. So this is how the study of brain function went in the mid 1800s. That we would observe people's injuries and say, hey, you seem to have lost a good chunk of your short-term memory, and your hippocampus is damaged. Hippocampus must have a lot to do with creating those new short-term memories. Things like figuring out Wernicke's area, part of your brain that has to do with spoken language, or Broca's area about producing spoken language. That's how we figured out that these different parts of your brain have to do with understanding and actually producing language. They're different parts, even though those are really related concepts, obviously. So problems with this way of studying the brain, well, you have to wait for people to have a particular type of brain damage. So over time, we learned a lot through this manner, but it doesn't allow like really focused or intentional study. It's very reactive. And you have to really wait, for the most part, until the patient has died to really understand the extent of the injury. So it's a very long-term, sometimes, sort of process of studying this. A way that you might get around this that would be very unethical in a lot of senses is to actually produce brain damage yourself. So to actually hurt someone's brain. So obviously we can't do this in humans, and frequently people do this with lower animals like rats, and I think people understandably have ethical objections to doing that in other animals too, but it does happen. Stimulating, overstimulating, or removing or damaging a part of a brain in an animal might tell us something about the types of function. But again, that involves leaps in logic because their brains are not exactly the same as ours, of course. Something you might do instead would be to stimulate the brain. So stimulating a particular part of a brain to create messages where there wasn't one naturally. This is, again, often done with animals instead, because it's easier to, put some sort of stimulation apparatus into an animal's head than a person's. Again, this might be unethical in some ways too, obviously, but it is done. So you can send an electrical charge or send an electrochemical message into someone's brain, and notice that, hey, if we stimulate this part of a rat's brain, they associate this lever with pleasure. So that must be the pleasure center of the brain. Again, not a super fine-grained way of doing this. And you can do this with humans. It can be done when there are certain types of brain surgery going on, because your brain does not have pain receptors. It doesn't have sensory neurons inside of it. So you can kind of poke around in a brain without hurting somebody, as long as you're not, damaging structures, basically. But again, this is not a very flexible way of doing this. Instead, we might listen to or look at the brain in different ways. So one way we listen to the brain is by something called an EEG, or an electroencephalograph. This is measuring just the entire electrical output or activity of the brain. So it's not looking at specific spots, but it's looking at how much electrical charge, basically, is going on in your brain. So an EEG output might look something like this, where you're measuring the total type of output going on from different receptors, where each one of these lines might be like, you've seen pictures probably of those little suction cups on somebody's head, or a cap with different electrodes coming out of it. That might be the electrical activity from each one of those individual electrodes on a cap or a suction cup array. So that's useful, but it doesn't help us figure out where specific parts of the brain are being active or taking a role. So to do something like that, we might use something like a CT scan or computerized tomography. So this creates instead a 3D picture of the brain, telling us something about its shape and structure. But This only tells us about what the brain looks like on the inside, really. It doesn't actually tell us the activity that's going on in it. So we can see when someone perhaps has an enlarged or damaged part of a brain, and that's a little easier to figure out if they're, have some kind of aberrant behavior, but it doesn't tell us about the activity. Luckily, we have some things that will tell us both of these things at once, both the activity and where the activity is happening. So something called the PET scan, positron emission tomography, accomplishes this. So the brain works by basically not digesting, but like using up different sugars. And if we can tell where the brain is using different sugars, we can tell where it is more active. So people will consume this harmless radioactive substance that has sugars in it, and that gets into the brain, and then you can use this PET scan, PET scan, to see where more of that radioactive material is being consumed. So that shows you where that substance is distributed in the brain over time. So that might look something more like this, where you see more radioactivity in this particular part of the brain, and you can conclude that there's more activity going on there. But an even better way of doing this, or more widely used way of doing this, that doesn't require someone to consume anything, is by using an MRI, or more specifically, an fMRI, which I'll talk about in a minute. So in your brain, you have all these different atoms that are behaving in a systematic, specific way when you send magnetic fields or radio wave pulses through them. So you can tell where the brain structures are and if they're moving and if they're active by sending these waves through the brain. So if you use this, you don't have the chemicals you have to use, and you can produce 3D images, which are much easier to use than the images from a PET scan. So this tells us something about the state of a brain, and if you take a bunch of MRIs over a short period of time, you can produce a sort of video almost of the brain function in a 3D way. And that was the F part of the fMRI, a functional MRI, showing the functions of the brain over time. So an fMRI is building by looking at the different blood flow and how things are being consumed in the brain. An MRI is just looking at structures. fMRI is looking at activity. So if you use several fMRI scans or several MRI scans in a row with alternating periods of rest, or having people do like math problems or looking at a particular color or something, then you might be able to see these differences in activity over time. Something like this. This might be someone who is asked to do a particular task, like remember something, or to think about a particular thing, or think about language in a particular way, and we can see where different activity basically lights up inside someone's head. Another way you might look at it is like this. It depends what programs you're using. It's going to look different in different programs and operating systems and stuff that you have, and how you display the information. But this is showing essentially the same types of information, just highlighted a bit differently. So this is how we learn quite a bit about the functions of the brain and how things are distributed in terms of different functions and labor across the brain. So I've talked already about the hindbrain. The brain stem and cerebellum are more primitive, things like motor coordination and keeping your lungs and heart and stomach working properly. The midbrain helps you coordinate all these functions with higher order things. It's sort of a communication station between the hindbrain and forebrain. But again, the forebrain is higher mental functioning, particularly that cerebral cortex that's kind of the outer foldy, wrinkly layer that we think about usually when we talk about brains. We do, like I said at the beginning, have a lot of evidence for the different functions in like the forebrain, the temporal lobes, the parietal lobe, and the occipital lobes. But I don't want to give the false impression that only memory happens in the temporal lobes, or only visual processing is in the occipital lobes. It's where these things are centralized, surely, but the brain works as a unit. It's, that old idea, you're only using 10% or 3% or 1% of your brain. That's crap. your whole brain is working all the time to accomplish the things that you're doing. And those, like I said, those distributed networks, those patterns of activation throughout your brain, they're working all the time to accomplish all these functions, not like only remembering things in your parietal lobe, for example. So really important to remember this. But it is, again, important to remember that we do have these divisions in some way, that it is working as a unit, but they specialize. It's like an organization that needs all of its parts to function properly, but there are specialties or different centers of where these things happen. So your cerebral cortex, again, has these two separate hemispheres, left and right, and each of them do have more specialized functions. Like I mentioned, your left brain has generally more language processing going on in it. And it's really always kind of bizarre to think about how your right side of, wow, I did the wrong one. The left side of your body is controlled by the right side of your brain, and the right side of your body is controlled by the left side of your brain. This includes your vision too, so this makes for kind of interesting things that we'll talk about again in sensation and perception. But so the things in your right visual field are being processed in the left part of your brain and vice versa. So there's these crossing paths in your optic nerves to how your brain is processing different information from your brain. Again, your corpus callosum, that bundle of fibers in the middle, connects those two hemispheres. And if it's removed or people are born without it, then we might see that they have particular deficits. Like if you had language information in the left side of your visual field and it was only being seen by the right side of your brain, you would have a harder time processing that language information because it's not going to your left hemisphere that processes more language. So again, we'll talk about that more deeply when we get to sensation and perception. And again, it doesn't mean you're totally losing these things, right? There's lots of overlap and contingencies going on in your brain. Okay, the last video, I'm going to talk about plasticity, genes, adaptations, and a little bit about the endocrine system.